So thank you very much for the nice invitation. So the only Greek word I can say this morning is kalimera. And I thank the organizers for inviting me to share some ideas on breast milk and diabetes prevention. So milk is a research topic I'm following about for 10 years now. I came to this topic as a dermatologist because I was interested in the effects of milk in acne, because if adolescents drink a lot of milk, the acne worsens. So my idea was there must be some signaling component in milk, so what is going on? And so I stepped into the milk business. So I'm not actually not an expert in breastfeeding and all the practical things, but I may share some theoretical ideas concerning milk and milk signaling pathways, which are quite important, I think, to understand the whole business. So breastfeeding, as you know, is what the World Health Organization says is the ideal food for infants. And I felt already that this is a wrong description, because it is, of course, a chance to prevent diabetes, what World Health Organization also says, but breastfeeding is a wrong concept. We are not feeding the infant, we are programming the infant by the breast. So the breast and the milk is not a simple nutrient. And I think this is the most important message at the beginning. This is mistake one, to think about breastfeeding as a nutrient for the infant. Of course, it is macronutrients and everything, and energy that the infant needs, but there's much more hidden in the signaling components of milk. And I think we have a devastating error in medicine, and it was introduced by Professor McKim-Merritt in St. Louis with a German guy called Schönthal, who developed the first formula made from evaporated milk protein in St. Louis, USA. And this professor said, there is nothing mysterious and sacred about breast milk. It is just food. And it is not. This is wrong. This is an idea. I think this was a boost for the artificial formula feeding. And this was a big run then on this business, because this prediction was made in a wrong way, because the understanding today of milk is much more than a food. It is a programming system. And if you just go into biology, then we know most components of the milk are produced under control of the human lactation genome. So it's the mother's genome which is transmitted over generation, from generation to generation, giving the milk to the infant. And the milk is only the transmitter of the signals which are created in the genes of the mother. And then we have the two functions, the dual function. We have the nutrition, of course, and we have a programming, and we will step into the programming processes of milk. And this is really fascinating what is going on. And there are more and more other colleagues like Thomas Rösner. They say that mother-child signaling through breast milk molecules or biomolecules. And today we will speak especially about the RNAs of milk, the exosomal RNAs, and I think this is quite interesting. So the evidence, is there evidence for a diabetes preventive effect of breastfeeding? We know we have the diabetes. The diabetes is now our burden of all nations. So we have increasing numbers of diabetes, and when we can prevent diabetes, we will do all that what we can, because this is a very cost-intensive disease and a real burden for humans. And why don't we use the signals at the beginning of our life to prevent this disease? So the primary prevention by breastfeeding is a good idea, and I don't go through the slides, but I only want to show there is a lot of epidemiological evidence from the last decades that breastfeeding prevents diabetes type 1 and type 2, but today I will specialize on type 2 diabetes. There are some reviews that breast milk is much better than formula in the prevention of breastfeeding. There's also a nice review from your country that showing all the data of that time. And so if you look into the epidemiology, then you find breastfeeding is advantage for the prevention of diabetes. And finally, this was also summarized by the Lancet breastfeeding group, saying that breastfeeding might protect against overweight and diabetes later in life. It's a little bit not so strict key message, because the data which are available point very strong to this diabetes preventive effect. So the baby is exposed to either the breast milk or a human formula. The baby cannot decide which type of feeding or programming it will get, but later in life it may ask, does breast milk deliver diabetes preventive effects that I miss in my formula? And I think this is a very important question. So we expose the baby to artificial systems which we do not understand and which are not evolution's preparations for the right programming at the right time window. So when we talk now about this signaling system milk, then we go into very exciting areas. So the milk delivers millions or billions of extracellular vesicles secreted by various cells of the milk, especially mammary gland epithelial cells. They are the big milk fat globules which transport most of the triglycerides and energy to the infant. There are other medium-sized extracellular vesicles which contain a lot of biomolecules. But today I will focus on milk exosomes. The milk exosomes are the smallest of these whole extracellular vesicles. They are in the range of about 100 to 150 nanometers. So this is the range of coronavirus, and the breast milk delivers per day 1 trillion of these particles to the infant. So these particles are very important, and there is more and more evidence that exosomes travel through the gastrointestinal tract, so they sustain the harsh environment of the gastrointestinal tract, and they reach the systemic circulation. I will show you some of these data. So milk exosomes have been characterized by the Karolinska Institute by Charlotte Admürel. They were the first to discover tiny particles close to the border of mammary gland epithelial cells. They are analyzed by electron microscopy. This was the original first paper, and they connected already these particles to immune development of the infant, and we just reviewed this data recently. Important for the information is that the exosomes are carrier systems of a lot of biomolecules. And they have a bilayer membrane, as you see, and this is important for their resistance against attacks in the gastrointestinal tract. And as we know, all cells of our body produce exosomes. So exosomes are produced by T lymphocytes, by liver cells, by brain cells, neurons. Every cell can produce exosomes, and exosomal traffic is now important to understand the connectivity of the organs, the traffic of information between cells, and so on. One of the ingredients, which is now the major focus of this topic today, is RNAs and special microRNAs. I will just introduce into this business because it is a little bit complicated, but it's important for the whole understanding. So if you analyze the exosomal microRNA content done, for instance, here by different authors, then you find there are a lot of very impressive, good expressed microRNAs. And I focus today especially on one because we cannot talk about all. And no human brain could just make all predictions with all microRNAs. So just we will focus on the dominant microRNA of breast milk. This is the microRNA called 148A3P. This microRNA you see here is highly expressed in all milks of all mammals. So it's the most dominant microRNA of milk. And these are data from the Israel group of Professor Golan Gerstel, showing it's also in skim milk, but it's also second in the human breast milk in the lipid fraction. So in the lipids and in the skim milk, you have this abundance of this microRNA in the native milk. So this is important information. That's why I just thought to go ahead with this microRNA in my concepts. Just a short reminder for you, what is microRNA really doing? A microRNA is, of course, synthesized by the gene. It's encoded on the DNA. And the microRNA is then processed. We don't go through all steps, but important is the final action. The microRNA in the RISC complex binds its corresponding messenger RNA. So each microRNA has by hydrogen bonding finds its sequence or seed sequence for a specific target messenger RNA. So when a microRNA has a good binding for the messenger RNA, then the RISC complex degrades this microRNA and RNA is destroyed. So this information on this messenger RNA is no longer be processed. So a microRNA is thus an extinguisher of messenger RNA signaling, and about 60% of our genes are controlled by microRNAs. So it's the most important regulatory system in our body, in our systems, and so microRNA signaling is something very, very important in the function of epigenetic regulation. Together with my colleague Gerd Schmitz, we published some years ago an idea that mammary epithelial cells abundantly synthesize, produce this microRNA 148A, which is then excreted by the exosomal pathway. The gastrointestinal tract cells can take up this microRNA, already exerting function in the epithelial cells of the neonatal intestine. But then we predicted that the microRNA may travel through the system, reaching all peripheral cells of the body, so doing signaling in the whole tissues and organs of the infant. So do we have evidence for this now? So because it was an early prediction, and of course, in the meantime, we have evidence, and this is one evidence is provided by the group of Professor Zempleni in the USA, and they gave bovine milk exosomes to mice. They labeled the exosomes with a special fluorescent dye, and then they looked after oral gavage of these milk exosomes, what happens and where they accumulate. And they found that these signals, milk exosomal signals, fluorescence, was found in most organs, and very special was an accumulation in the brain. So this means that the breast milk exosomes have a contact to brain development or neonatal development, and I think this is a very important information, especially in terms if we speak about later on not on breast milk, but cow milk and maybe glioblastoma development. This is a very interesting issue we could follow. There is further evidence, and very recent evidence, by the group of Philipp Weil. This is close to my hometown at Wittenherdecke University. They made a big study with artificial microRNAs they gave into breast milk and milk exosomes, especially cholesterol exosomes, and they looked where is there really a passage into the through the gastrointestinal system, and is there a chance for the regulation in the system? And they approved that this is really true. So we have a very recent information and confirmation about the uptake and systemic availability of milk microRNAs. So now, if you look to the data we have, the breast produces about 180 trillion milk exosomes per day, delivering this to the child. So this is an unbelievable high number, and this is not, I think, done for feeding. This would be absolutely ridiculous if milk exosomes were just made to produce these complex systems as a feeding system. This is a messenger sent to the infant, reaches the blood and the circulation, and there are a lot of other papers you find here which support the idea that breast milk exosomes and their RNAs are bioavailable. And there's a recent review also showing this cascade. So the breast produces these milk exosomes with their exosomal function, and they are absorbed in the gastrointestinal tract. They already have an impact on the gastrointestinal tract cells of the, on intestinal maturation, of course, and they reach the systemic circulation. And if they reach the systemic circulation, we have also to think about, do they reach the pancreas and the beta cells of the pancreas? And this is the next idea. Exosomes from various tissues communicate with the pancreas. So the pancreas, we know already, is highly regulated by exosome traffic. So, and the pancreas is also very peculiar in a way that the blood epithelial cells, which are surrounding the beta cells, have very wide windows, fenestrae, which are in the range of 100 nanometers in size for the uptake and interaction with macromolecules. And this is why exosomes can reach the beta cell, so the anatomic situation is available. And we now have information that extracellular traffic is a very important functional role for beta cell function and viability. And we see the pancreas can be stimulated by exosomes derived from various organs, like the liver, like the muscle, like the adipose tissue. When this is the case, then we can predict that also breast milk will affect the beta cells in the pancreas. We have these data which are very recent, that, for instance, that in obesity, milk exosomes have an unfortunate effect on the beta cell. They stimulate proliferation because the beta cell has to produce more insulin to maintain these extra adipose tissue. So there are immune cells, they can, via exosomes, communicate with the beta cells. And of course, if we do a lot of sports, we produce also exosomes which are favorable in controlling beta cell homeostasis. So this is already known, so that exosome traffic is important for beta cell homeostasis. So then we have the next first important new hypothesis. Do milk exosomes have a direct impact on the beta cell? I showed you that there is already a lot of traffic, and we have to assume that during the neonatal period, the breast produces exosomes. And I told you, the breast produces 180 trillions per day of exosomes. There will be exosomes traveling to the beta cell. And as the beta cell is highly dependent on exosome traffic and regulation, this should be a physiological route during the postnatal period. So this is another review showing that exosomes really travel through the intestine, reach the circulation, and these authors already discuss an impact of exosomes on various organs and, of course, the pancreas. So now what is characteristic for the neonatal postnatal beta cell? The postnatal beta cell is a very proliferative tissue and has a very high mTORC1 mechanistic target of rapamycin complex 1 activity. mTORC1 is a key controller of anabolism and proliferation. It's a kinase with a central position in the cell. And the AMPK is low during this time of proliferation. The AMPK is a kinase very important for function of the beta cell and insulin production and also for the energetics of the beta cell mitochondrial function. So in the postnatal period, we use the time for beta cell proliferation with mass expansion, but at this time the beta cell has a reduced glucose-stimulated insulin secretion. It's functionally immature. The beta cell of the neonate is immature because it does not need to respond to varying glucose levels. It is maintained by lactose, which is, of course, over lactation, mainly in a similar concentration, is provided in a similar concentration. But after weaning, then the infant has to respond to changing glucose levels. So during the lactation period, the beta cell maintains proliferation to produce a good beta cell mass and have a good beta cell mass expansion. And this is then an important function for diabetes prevention for the whole life, to have a good proliferation, a good pool of beta cells. And we know that the neonatal period is a time of cell proliferation in the beta cell. And in the normal situation, this declines, proliferation declines. Of course, there is some proliferation in adolescence, but with normal aging, it declines. And in type 2 diabetes, we have again an increase, and also in type 1, in proliferation. So this is important. Keep this in mind because we will discuss this later. These are data from rodents and also is known for humans. For me, the most exciting paper of the last years was in the Journal of Clinical Investigation 219 by Jafar. And they found that with the weaning, there is a switch from high mTORC1 activity to increased AMPK activity. And you also may remember that AMPK is the enzyme which is activated by metformin, our most important anti-diabetic drug. So there is a switch. So you see, neonatal period, the activated AMPK is low, and with in the adult beta cells, the AMPK is high. So this is a switch, and with this switch, the beta cell becomes operative, functional, and is able to produce glucose-stimulated insulin secretion. So hypothesis 2 is then, do milk exosomes-derived microRNAs, the cargo of the exosomes, modify genes of the beta cell? And do milk exosome-derived microRNAs enhance mTOR signaling, what has been shown, and suppress AMPK? Can we show this by genetic or by a matching of microRNAs with their target genes? And that's what I did. I looked into the genes which are important for mTOR signaling and AMPK, and looked it up. Do they match and how they match for the most important microRNAs in the beta cell? especially we focus on microRNA 148A. All right, so this was then the prediction. We say human milk exosomes are delivered, potentially delivered, we have to say, to the exosomes of to the beta cells. And would they increase beta cell proliferation by increasing mTORC1 and decreasing AMPK, just associated with a reduced glucose-stimulated insulin secretion during the neonatal period? What is the evidence? So we have the situation that during breastfeeding, we have, of course, a supply of milk microRNAs. And with the weaning, the supply of milk microRNAs declines. So it's not a switch like on-off. It is a slow situation, but the breastfeeding declines. Other food is introduced, but of course the number of milk exosomes will go down, and this will be, I think, the signal for the appropriate beta cell differentiation then to lower mTORC1 activity and higher AMPK so that the beta cell gets functional. So this is what I already summarized from the IBFAN data. So the beta cells switch from high mTOR to higher AMPK during the postnatal maturation. This has been shown in these rodent experiments. And the AMPK finally triggers oxidative phosphorylation. And this is important for the energy production of the beta cell and is closely related to insulin secretion. And the weaning is the critical switch from high mTOR to high AMPK in the beta cell. And in type 2 diabetes, and this is a fascinating thing, we have a situation reverse back to the neonatal period. Type 2 diabetes in adults, we have again increased proliferation, but of course less function, less maturity. So at the moment, the researchers in the diabetes field, they say type 2 diabetes is a de-differentiation of beta cells back to the neonatal phenotype. So if we drink milk and going on with this for lifetime, we may have adverse effects on the system, because when we always have a stimulus for proliferation of the beta cell, then we will have earlier beta cell apoptosis. The cells will die earlier, and we will have a functional deficit with time. So if you look now into the possible genes, the milk microRNA will change. For instance, the milk 148A extinguishes expression of tuberin, TSC2, which is an important inhibitor of the mTOR pathway. We have other important links. We see that microRNA 148 suppresses at least two components of the AMPK enzyme. So it's downregulating the AMPK, we can show that. And also DNA methyltransferase 1 is suppressed. And this is important because this would lead to an upregulation of transcription factors like E2F1, like CMYC, and the epigenetic regulation will lead to a hypomethylation of the H19 gene. This is an important long-coding RNA. H19 is highly expressed in fetal tissues and also in the postnatal period. And H19 is highly expressed in the proliferating beta cell. So this is very important because when the beta cell matures, H19 expression goes down, and this is associated as well with the functional maturation of the cell. So if we provide this milk exosomal traffic, you can upregulate H19, and as you may know, H19 is highly expressed in most of our solid tumors like mammary cancer or prostate cancer because it is a program of the neonatal fetal area, which is upregulated in stem cells, cancer stem cells. So this has been shown by the group of Regazzi here, that the growth stimulatory effect in the beta cell upregulates H19. And this is important. H19 is a long coding, a non-coding, but it's a long non-coding RNA, and this can quench LET7 microRNAs. And LET7 microRNAs, they can inhibit proliferative signaling pathways. And if LET7 is quenched by H19, then you activate proliferative pathways. We don't go in all the genes, but everything fits well together. So the microRNA 148A3P attacks a lot of key genes in the beta cell, and we can explain, especially by looking up to all these genes in particular, that the beta cell function is reduced, the proliferation is increased, and we have clear evidence that this fits very well together with the observations of Jafar et al., who found this switch of mTOR to AMPK during weaning. So something is wrong when we give infant formula. So if we give infant formula, we see already, this is a nice paper, that breastfeeding has a totally different expression pattern, for instance, of PPAR, PPAR gamma, PPAR alpha, FTO gene. The FTO gene, for instance, in breastfeeding, you see this, blue, is very low. FTO is fat mass and obesity gene. So the type of feeding controls the expression of key genes for obesity. This is only a little link so that we see with a formula, with a total breastfeeding, you have the best protection in terms of FTO. And with the exclusive formula feeding, you have very high 10 or more times more FTO expression. So it's absolutely not the physiological axis, and this is of critical concern, especially also if we look into diabetes development. So what happens now when the beta cell comes from the proliferative state into the functional maturation? And in the diabetes research, they know already there is one important transcription factor called estrogen-related receptor gamma. And this is a key regulator of glycolysis. And this is suppressed, and it is a target gene of microRNA 148A. So this is another important regulator. And when the microRNA 148A is extinguished, the expression can go up, and this then controls the functional maturation of the beta cell, especially mitochondrial activity and oxidative phosphorylation, which is very important then for glucose-stimulated insulin secretion. So we can really predict that with the fading of milk exosomes, we have a maturation of the beta cell to function. And there is another important transcription factor which is controlled by this microRNA. It's PPAR gamma coactivator 1 alpha. It's also a target gene of the milk and the milk signaling. And this controls, for instance, also the expression of estrogen-related receptors. We see there is a whole network of signaling connected to this microRNA, and this transcription factor, PGC1 alpha, is also a dominant transcription factor for mitochondrial function. So there is another little outlook. We know that we are controlled by circadian rhythm, by genes which we say controlling our internal clock, which is actually the day-night rhythm on Earth. We have 12 hours light, 12 hours darkness, just in short. So our genes and our internal clock of our cells is controlled by clock genes. And I don't want to go too deeply because of time into this topic, but it's important to realize that these clock genes are also controlled by the microRNAs. For instance, I told you that PGC1 alpha is controlling the expression of critical clock genes in the cell. So we can say that also the microRNA 148A might have an impact on our circadian clock. And this means the baby has another clock. It's not the day-night rhythm we adults are in, because the baby also is feeding, I think you know it better, 3 hours, comes to the breast again. So it's another rhythm, and it makes sense that milk disrupts the circadian signaling, and we know already that these circadian clock mechanisms are closely linked to insulin secretion and to glucose homeostasis. So important, again, this important gene which controls mitochondrial function and the circadian clock is controlled. You can see this on target scan and other libraries. It's controlled by this microRNA 148A. Also shown in a pig model by a Chinese research group recently. So extracellular vesicles then control also our circadian rhythm and synchronization. This is important for insulin secretion, and there are specialists in the field of diabetes linking the circadian rhythm to diabetes. For instance, shift workers, like in medicine or in pilots, changing always time zones. They have a higher risk to get type 2 diabetes in their life, so it's increased. So the clock genes, of course, are controlled by microRNAs, and also there is a link to the H19 and the control of clock gene homeostasis. So this means that weaning is a special time. Weaning is a time to make the beta cell mature, and this is already published by other groups, not related, unfortunately, to the breast milk, but to the beta cell microRNA content they found. And one interesting thing is also that the clock genes, one important microRNA is a microRNA called 17.5p, and you see in the postnatal period it's found to be much higher than in adults, and this microRNA controls clock genes, and this is also a microRNA found in breast milk exosomes, especially in the exosome fraction. This microRNA is highly expressed. So the microRNA 117p is reduced by ultra-heat treatment of cow's milk. And this is interesting. It's high in raw milk and in pasteurized milk, but it's destroyed by this by higher heat treatment. And it has been shown by the group of Zhang that there is a loss of bioactivity of microRNAs if you heat the milk, especially ultra-heat the milk, but there is a survival of the microRNA system by pasteurization, which is only 78 degrees. It's not enough to disrupt the exosome membranes. And there is another change shown for microRNAs during the postnatal period and adulthood, which can be explained by the high expression of microRNA H19. We don't go to this into detail, but it's now clearly recognized in the field that there's a circadian control of beta cell function, and we have to think about how breast milk and breast milk exosomes modify the system. So we can now say, okay, does disturbed postnatal microRNA 148A signaling enhance the risk of diabetes later in adult life? Okay, there is an absence of this microRNA in formula. We know this. There have been a lot of measurements. The formula does not contain exosomes. So there are reduced levels also in the milk, in the breast milk of obese mothers. Very interesting. And also the milk, the breast milk of mothers with gestational diabetes have also a reduced level of this important microRNA. And now, but what in my opinion is a very critical observation, that also caesarean section reduces this important microRNA. So now we go on and think more. Okay, I make it short because the evidence has been published. Because this microRNA is not found in formula. Formula is microRNA deficient. There is no functional microRNA in formula shown by Professor Zemplini's group. There's the interesting work on obesity and relation to milk exosomes. And you see the microRNA is significantly suppressed in the breast milk of obese mothers. There's the same group showed that also in gestational diabetes we have a decrease in the breast milk concentration of this microRNA 148A. And caesarean section modifies the concentration of this microRNA. I think this is found in mature and transition milk, was a statistical evidence. So when we do our business in the perinatal periods as medical doctors, we give milk to our mothers, which is an unusual situation. No adult gets milk. Then we give milk. Then we provide a natural section. So if we deliver by caesarean section, and which I realized is in your country about 50%, this even more, then you disturb regulatory circuits. I think it's the right for the child to be born on the right way. The group of Regina Golan-Gerstel from Israel, Hadassah University, they checked the expression of this microRNA in relation to oxytocin stimulation. And you all know that oxytocin is the most important hormone, highly upregulated during the process of delivery. And if you stimulate cells, breast cells, with low levels or high levels of this important hormone, you see you have less or more significant, more expression of this microRNA. So this means that caesarean section has an impact on the quality of the breast milk. This is other data I found from your country. You say it's even more. I found this data for the high number of caesarean section. And it would be nice, I think, to make more research on, I have no data, but we should really look up a correlation between caesarean section and type 2 diabetes. I looked at only up a little briefly, but I think countries with high numbers of caesarean section may have higher risk of diabetes. It's too early to prove, but it's important. So do bovine milk exosomes, last talk, part of the talk. The bovine milk exosomes are very similar to the human milk exosomes. The bovine milk exosomes have the same microRNAs, and milk microRNAs are a very archaic system. The microRNAs, the sequences, are nearly identical. For instance, I'll show you for this important microRNA, it's absolutely identical. So when we now take milk exosomes during our adult life, when milk is normally forbidden to be taken in in a native form, then we may de-differentiate the cells back to the high mTOR, low AMPK phenotype. And look to the cow. The cow has to produce much more exosomes than humans. It's 10 to 1,000-fold higher, the numbers. So this means that the milk exosomes in cow milk, in native cow milk, is of course raw cow milk, the highest. You're losing them by pasteurization, but not so much by ultra-heat treatment. You destroy them. And by cooking, I think, also. It depends on the time. But we are exposed in our modern world. We have a refrigerator. We have the fresh milk in the refrigerator. We can open the door and have every day and every time access to an evolutionary signaling system which is normally prohibited for adults. Look, these are the sequences of the microRNA 48 3P in the bovine system and in the human system, absolutely identical, so they can match our genes. So in our Western world, with our technology, we are controlled by the cow. There is a Greek, what is called on Crete, I think biochemically we are minotaurs. Okay, so what's going on? So pasteurized milk, we know, transmits microRNAs. We have a loss of microRNA by ultra-heat treatment, but we have a supply by fresh milk, pasteurized fresh milk. So what happens then? So when we have the normal situation, the child gets breast milk, very nice. The milk exosomes promote beta cell proliferation. This will be a good ideal situation, a good starter for life, to have a good pool for beta cells. And this is then a very good process. It terminates with weaning. And then we start again with a supply of bovine milk exosomes by pasteurized milk. And this could then de-differentiate the beta cell back to a higher proliferation status and to less maturation, to less insulin secretion, to beta cell de-differentiation to the postnatal phenotype. And this kills the beta cell in the long term, because if you have always proliferation, you have the risk of earlier apoptosis and senescence and, of course, beta cell failure. So that's what, in my opinion, happens in our Western world. With the bovine milk exosomes, we promote again beta cell mTOR activity and reduce AMPK. And this idea is published recently. We have published this recently with my dear colleague Gerd Schmitz in the International Journal of Molecular Science. Yeah, Sciences. So this is really true. If you look into the paper of Jafar, they showed the evidence that in type 2 diabetes, the AMPK is low again, so it's going down, and the S6 kinase is an indicator of high mTOR activity. It's downstream of mTOR C1 is high. So we have indeed de-differentiation back to the postnatal period. And if you look into epidemiological data like the Lifeline cohort studies in the Netherlands, they looked up different milk products and diabetes relation. And they found a relation between milk consumption and diabetes, but not with fermented milk products. So you can go on, I think, and eat your Greek yogurt because fermentation kills also the exosomes. The exosomes are phagocytosed by the bacteria. So the lactation products over our last 10,000 years, and we all know It started all in Turkey, in the area of Tigris, and what we say is the start of the dairy. But at that time, there was no refrigerator. We had fermentation was a normal process. So cheese is fermented milk. Of course, it's the protein of milk, which is very valuable, but too much leucine amino will also activate mTOR. So if you have too much milk amino acids, like also too much meat, then we drive the mTOR system. This is, of course, another story. But if you look to the microRNAs, this system was destroyed by the process of fermentation. And fermentation, we switched with the pasteurization and the refrigerator to another exposure of humans with a full power of mammalian evolution. So what we did, final slide, we just changed our evolution in terms of milk signaling. The normal signaling, breastfeeding of milk, microRNA signaling is a time of the postnatal period of lactation. Controlled by our own lactation genome, not controlled by the laboratories of the food companies, controlled by our genes over millions of years, and I think we should rely on our genes and not on artificial systems. What we do in fetal life, we expose our mothers with the bovine exosomes. The gynecological societies recommend milk and dairy consumption during pregnancy. They do not differentiate between the milk types or subtypes, so they have to take off fresh milk from their recommendations. They only think in terms of calcium, which is a little bit stupid, I think, only to think on such a simple compound, which is also in other vegetarian products. I think this is not a good thing. The group of Professor Zempleni showed if you give milk exosomes to pregnant rodents, you find also the uptake of the exosomes and they increase fetal growth. And this is no good. You have the problem with macrosomia. We have more and more babies which are too heavy at birth, and then they get cesarean sections because they got too big. So we destroy already the fetal axis. And then we interfere with a normal birth. And then we interfere with a type of signaling programming because we give formula feeding. So we made everything wrong we could. And then we wonder why we have so much diseases of civilization. And then we corroborate this by giving milk in a time period when milk is not allowed by normal lactose intolerance. This is our original gene. We are mutants, we know, but 10,000 years ago we were not mutated. We were mutated because we came into the milk exposure 10,000 years or 7,000 years ago. But we just changed our evolution. We are now we changed top and we stay no more of sound ground. And I think we should just realize we have to come back to our original roots and to our secure mammalian genome of lactation. Thank you very much for your attention. I think I skipped. Should I give a summary? All right. Then it's easier for reading, I think, for you. So breastfeeding is not a simple nutrition, but the execution of postnatal signaling controlled by the human lactation genome. Breast milk-derived exosomes and their microRNAs are the key regulatory transmitters that reach the systemic circulation of the infant. And it's not only microRNA, it's also long-coding RNAs, other RNAs. It's much more complex, but it's only the tip of the iceberg. Various exosomes communicate with beta cells, this is sure, implying the existence of milk exosome traffic to the beta cells. So it's also important in terms of milk bank So if we would have, I think it's very good to have a milk bank and to supply human breast milk. Absolutely important. In Germany, before we got reunited, we had in Eastern Germany, we had milk banks. In Western Germany, we did not have. So in East Germany, they had less incidence of allergies. We could not understand. But in West Germany, we had the better technologies, we thought, but we had more allergies. What happens? MicroRNA 148A controls DNA methyltransferase 1. So it's suppressing this DNA methyltransferase, which is an important step for the maturation of regulatory T cells. So the regulatory T cells control the allergy business. So in the eastern part, they had a good development of the immune system, and in our western part, we had formula, and we had our disaster with allergy. Now we have, after the wall was opened, now we have the same numbers in both sides of Germany. So this system is also important for the whole immunology business, but I cannot go into detail because the topic was diabetes. So during the postnatal lactation period, we have, of course, mTOR-driven beta cell proliferation. This should be maintained by breastfeeding. So the mother is doing the best for her baby, for diabetes prevention, I think also for obesity prevention, cancer prevention, by providing her breast milk, her own gene-developed products, and is doing the best. It's not only a decision, what should I take? The problem is that in the society, people think it doesn't matter really. It's breast milk or the other milk. Both look white, and the child on the first look develops, grows even a little bit earlier than with breastfeeding, but the biochemical situation is hidden, invisible, and not recognized. Mechanistical evidence supports that milk exosomal microRNAs control beta cell proliferation, also induce glycolysis, or glycolysis, how do you say? It's because beta cell needs glycolysis for proliferation. All tissues, which also cancer tissues, use glycolysis for fast proliferation, and this is also the beta cell is doing this, but this has to, then has AMPK has then to be downregulated, and this happens with microRNA 148A, and in later life, we want to kick up the AMPK again by giving metformin. So important is this because all this interaction with mitochondrial activity, circadian rhythm, glucose-stimulated insulin secretion, this fits all together. It's a signaling network which we have to look to. And weaning is then the critical step. And if you look in native countries, the mothers provide milk. They give then the first external food, again with the milk. So it's a continuum. The oral tolerance is induced by the milk, and also the whole signaling can be adapted. So the switch mTOR to AMPK will be physiological, and the beta cell then goes into maturation and function. And formula, of course, does not provide the whole system. It's totally deficient in the epigenetic regulation, and the epigenetic regulation in terms of FTO expression I showed you is a disaster. Maternal obesity, gestational diabetes, and cesarean section are all found to be associated with reduced microRNA 148A expression. This means this is better to give the milk of an obese mother, of course, because there is still microRNA present, but it's less than the mother would be lean. The gestational diabetes is a risk again. I think everything is interconnected again. And the cesarean section, we can talk about, I think for the hospitals, it's also very nice. It's like an operation, gives good money, and it is pleasant for the women, no pain. But we do not a good job when we continue with this manipulation of our perinatal physiology. So persistent bovine milk signaling continued then over decades. So you're drinking every morning your muesli or your cornflakes with fresh milk. Then you increase again beta cell dedifferentiation in terms of diabetes, but you also increase mTOR signaling in other organs. And we now have very good evidence from good studies that milk consumption in women for some years, prospective studies from Loma Linda University, California, they showed higher breast cancer risk for women drinking about 150 milliliters milk per day. It's a daily signal. It's not the milk once or twice a week. The daily signal seems, and it should not be too high. It's 1 glass or 1.5. It's enough to enhance cancer risk. And the same has been shown already for prostate cancer. And these are our major cancers in our Western world, clearly now related also to milk consumption. Yeah, I think this was a message. And who would like to read it in more detail, what I told you today, is can look it up in this journal, and it's free. It's freely available. It's open access. Thanks a lot.